everyone, I'm Navid Ahmad. Today we are discussing about the mechanism of digestion. Let's start with the introduction. Digestion is the process of mechanically and enzymatically breaking down of the food into substances for the absorption into the bloodstream. In the process of digestion, the macronutrients are broken down into simple molecules that can transfer to intestinal epithelium and enter the bloodstream for the use in the body. The food contains three macronutrients that are required in the digestion before they can be absorbed. They are proteins, carbohydrates and the fats. These are the three important macronutrients that are required for the body. Let's see the digestion of these three important proteins one by one. All the types of food molecules undergo digestion at different regions of alimentary canal like mouth, stomach and the intestine. These regions provide a suitable environment for the conversion of complex food molecules into simple soluble food molecules. So these regions have a different types of digesting enzymes that helps in the digestion of these food molecules. The first one is the digestion of the proteins. Digestion of proteins in the mouth. Proteins do not undergo any change as there is no protein digesting enzyme present in the saliva. Protein digestion in the stomach. In the stomach, proteins are digested with the help of pepsin and renin. The pepsin, it is secreted in the inactive form known as pepsinogen. It is activated by HCl. The pepsin act on the protein and converting it into proteoses, peptones and polypeptides. In the diagram, we observe that the zymogen, which is an inactive form of pepsinogen, which is activated with the help of HCl, and the action of HCl on the pepsinogen convert the pepsinogen into pepsin. This pepsin acts on the proteins and convert it into proteoses, peptones and the polypeptides. Renin or chymosin. It is a milk curdling enzyme. Renin acts on milk protein casein and converting it into para-casein. In the presence of calcium of milk, the para-casein is further converted into calcium para-caseinate, which settle down to as a curd. The pepsin act on the curd and converting it into proteoses, peptones and polypeptides. In the picture you observe that the renin act on casein and converting it into para-casein. In the presence of calcium, this para-casein is converted into calcium para-caseinate, which is settled down to form a curd. So this pepsin act on the curd and convert the curd into proteoses, peptones and the polypeptides. The protein digestion in the intestine. In the intestines, the proteins are digested by the following enzymes. The first one is trypsin. It is secreted as trypsinogen, which is an inactive form of the trypsin, which is activated by an enzyme known as enterokinase, so which is secreted by the intestinal juice. The trypsin is convert the proteins into proteoses, peptones and the polypeptides. So in the picture you have that the enterokinase act on the trypsinogen and converting the trypsinogen into trypsin. And this trypsin at a pH of 7.1 to 8.2, it converts the proteins into proteoses, peptones and the polypeptides. Now the chymotrypsin. It is secreted as chymotrypsinogen, which is an inactive form of chymotrypsin. It is activated by the trypsin. So chymotrypsin act on the proteins and converting it into polypeptides. In the picture you observe that the trypsin which convert the chymotrypsinogen into chymotrypsin. This chymotrypsin at a pH 7.12, 8.2, it converts the proteins into polypeptides. The third one is carboxypeptides. The carboxypeptides act on the protein and converting it into tripeptides, dipeptides and amino acid. In the picture we observed how the proteins are converted into tripeptides, dipeptides and amino acid with the help of carboxypeptides enzymes. The fourth one is aminopeptides. Aminopeptides act on proteases, peptones and polypeptides and converting them into dipeptides, tripeptides and amino acids. So in the picture we observe how the dipeptides, tripeptides and amino acid formed with the help of aminopeptides. And the last one is tripeptidase and dipeptidase. This tripeptidase and dipeptidase acts on tripeptides and dipeptides and converting them into amino acids. So amino acids are the simplest forms of the proteins. At the end of the digestion, so proteins have been completely digested into the amino acids. So they are ready to be absorbed. In the next slides, we observe the digestion of the carbohydrates. Digestion of carbohydrates. The carbohydrates are also known as carbs. They are the sugar molecules. Along with the proteins and fats, carbohydrates are one of the three main nutrients found in the foods and the drinks. So let's see the digestion of carbohydrates in the mouth. The food is masticated by teeth and mixed with the saliva contain the salivary amylase. The salivary amylase act on the starch and converting them into maltose. So in the picture we observe that the salivary amylase at a pH of 6.8 to 7.8 they convert the starch into maltose. The maltose are the disaccharides. And the second one is carbohydrate digestion in the stomach. The carbohydrate do not undergo any change in the stomach because there is no carbohydrate digesting enzyme present in the stomach. Now the carbohydrate digestion in the intestine. In the intestine, carbohydrates are digested by the following enzyme. The first one is amylopsin. It is act on starch and converting it into maltose. In the picture we observe that the amylopsin, it is amylase that act on the starch at a pH of 7.1 to 8.2 and converting them into maltose. The second one is maltase. The maltase act on maltose and converting it into two molecules of glucose. In the picture, the maltase at a pH of 7.1 to 8.2, it act on maltose and converting into two molecules of glucose. And the third one is sucrase. The sucrase act on sucrose and converting it into glucose and fructose. So at a pH of 7.1 to 8.2, sucrase act on sucrose and converting it into glucose and fructose. And the last, the intestinal enzyme is the lactase. The lactase act on lactose and converting it into glucose and galactose. So at the intestine, the pH range from 7.1 to 8.2. So at the pH of 7.1 to 8.2, the lactase act on lactose and converting into glucose and the galactose. And at the end of the intestinal digestion, the polysaccharides are converted into disaccharides and then disaccharides are converted into monosaccharides which are ready to be absorbed. And the last one is digestion of lipids. Lipids are the molecules that contains hydrocarbons and make up the building blocks of structure and function of living cells. The examples of lipids includes fats, oils, waxes and certain vitamins like A, D, E, K. Let's observe the digestion of lipids in the mouth. The lipid do not undergo any change in the mouth as there is no lipid digesting enzyme present in the saliva or the mouth. Lipid digestion in the intestine. In the intestines, the lipids are emulsified. The emulsification occurs by the action of bile salts. The bile salts act on the fats and break the fats them into smaller droplets of 0.1 mm in diameter. The gastric lipase act on the emulsified fat and converting them into fatty acid and glycerol. So in the picture we observe that the emulsification of fat take place with the help of bile salts. After the emulsification, the fats are converted into smaller fat droplets. The enzyme like gastric lipase act on the smaller fat droplets and converting it into fatty acids and the glycerol.